Hello everyone, my name is Renming Liu. I'm a PhD student in the Computational Mathematics, Science, and Engineering department at Michigan State University. Today I'll be talking about accurately identifying disease genes and disease contexts using context-specific gene interaction network embeddings. Identifying disease genes is a fundamental task towards better understanding of complex human diseases and the pathways or mechanism underlying them. This is of great importance because it could lead to potentially novel and effective treatment strategies for many complex diseases. And the problem of disease gene annotation is as follows. Suppose for a particular disease, we currently have some knowledge about the genes that are associated with it, colored in blue in this example. We would like to find out some other potential related genes in the human genome for this particular disease, colored in orange in this example. Many previous works have shown that gene interaction network is a powerful tool for studying the functional role of human genes. In particular, our previous study called GenePlexes, which is published on bioinformatics, have demonstrated the grand opportunities in using network embeddings to predict disease-associated genes. Despite some success in annotating genes using gene interaction networks, there are still some improvements to be made. In particular, modeling the human gene interaction networks under different contexts, such as tissue, can better take into account of the heterogeneous nature of complex human diseases, where there are several hundreds of genes are being involved. For example, when studying neurological diseases, it is more beneficial to use the brain-specific network. And when studying heart-related disease, it is better to use the heart-specific network. Thus, in this work, our main goal is to extend this idea of constructing networks, context-specific gene interaction network, to contextualizing network embeddings. Now, one straightforward solution is to directly take the context-specific embedding, context-specific interaction networks, and embedding them directly. However, we would like to have a more flexible and general approach that is independent on whether a particular context-specific network is available. We will do so by using a backbone gene interaction network that is context independent and contextualize the embeddings using gene expression profiles that define the context of interest. Before I go into more details about our context-specific embedding approach, I want to briefly touch on Notuvec, which is a network embedding method that we build upon. The general goal of network embedding is to represent the full network that is equivalent to an n by n matrix called the adjacency with an n by d dimensional embedding matrix, where the embedding dimension d is much smaller than the total number of genes n in the gene interaction network. This is very useful because a low dimension means we can train our machine learning model for the task more efficiently. Now, to learn the embeddings, Notuvec first generate random walks on the network, which are essentially sequences of nodes. Using these node sequences, it then learns to position every node in the network in the embedding space based on how frequently two nodes co-occur within a few hops of the random walk. The main idea of our context-specific embedding approach is then to guide the random walk towards genes that are more relevant to a particular context of interest, which are colored in dark gray in this example. I want to point out that in practice, we do not only have a binary characterization of gene context relevance, but rather each gene has a continuous value score that indicates how relevant that gene is to this particular context of interest. Finally, these context gene relevance scores are computed based on gene expression profiles using either mean expression or differential expression approaches. At this point, given a context-independent gene interaction network and k number of gene expression profiles, we can generate k different numbers of context-specific embedding spaces. But remember that our final goal is to generate disease gene predictions. To do so, we train a collection of logistic regression models using all context-specific embeddings. We then aggregate the predicted probabilities from all these models and weight them according to how well they perform on a validation set for this disease, resulting in our final ensemble model. We hope that if a context is appropriate, if a context is appropriate for a particular disease, using its corresponding embeddings would result in better prediction performance, and hence would be upweighted in the final ensemble model. In this way, we not only learn a model that is able to uncover disease genes, but also provide insights into contexts that are relevant to the disease. To systematically test our approach, we use BioGrid as the backbone gene interaction network and obtain gene expression datasets from publicly available gene expression studies and tissue-specific gene expressions. We then derive disease gene classification task from this GeneNet and evaluate the model's ability to uncover disease genes. As mentioned before, we would like to know whether our context-specific embedding approach can better uncover disease genes than the usual context-naive embeddings, and whether our ensemble approach is able to shed light on the disease contexts. To answer this first question, we compare the disease gene prediction performance using context-naive embeddings and our context-specific ensemble approach. We treat the gene expression studies and tissue-specific gene expression separately, resulting in two different context-specific ensemble. The disease gene prediction performance is measured by the log2 fold change of the area under precision curve over the prior. This metric is specifically chosen as it corrects for class imbalance issue and also focus more on the top rank predictions, which are practically more useful as gene association predictions. As we can see from this figure, both context-specific ensemble achieve significantly better disease gene prediction performance than the context-naive counterpart. On the other hand, we would like to investigate whether our ensemble approach is able to uncover disease contexts. In other words, do known contexts for a particular disease receive larger weights in the ensemble model? To answer this question, we first collected disease to study correspondence from previous manual curation efforts for a subset of diseases. We then transform the coefficients so that they are at a comparable scale and compare the transformed coefficients for the studies that are known to be associated to a particular disease and those for other studies. We can see that indeed, for a particular disease, the known disease contexts are being upweighted. Now let me show you that a couple of examples disease context inferred by our approach. Here, we used gene expression study as the contextual information and trained the model to predict the ulcerative colitis disease genes. You can see that the top two studies are directly related to the ulcerative colitis. Moreover, the fifth and sixth studies are also related to the ulcerative colitis as they are related to the colonic cancer. Similarly, when using tissue gene expression as the context information, we see that in many cases, the top predicted contexts make intuitive sense for a particular disease. For example, the heart tissue is related to the heart conduction disease, the blood tissue is related to the vein disease, and the brain tissue is related to the neurological disorders such as bipolar and anxiety disorders. In the future, we would like to take our approach one step further by generating cell type specific gene network embeddings using single cell RNA-seq data as our contextual information. In this way, it can help further understand disease context at the cell type resolution. We would also like to experiment with some other advanced ensemble methods and study their ability to uncover disease contexts as well. In summary, in this study, we aim to understand disease genes using network embeddings in a context-specific manner. We have developed an effective approach to generate context-specific embeddings and combine them with ensemble learning to successfully uncover both the disease genes and disease contexts. Thank you all for listening. I want to take these opportunities to thank both of my advisors, Arjun Krishnan and Matthew Hearn, for their great support and guidance. I also want to thank all the lab members that are also being extremely supportive. You can find out more information about the software we developed for generating embeddings and predict disease or predict gene annotations if you are interested.